Hello, and welcome to this video on how yeast digests sugar. As seen in previous videos, yeast converts glucose molecules into ethanol and carbon dioxide when in low oxygen conditions. This video will explain how yeast executes sugar metabolism. Yeast primarily utilizes glucose, breaks it down into pyruvate and then acetyl-CoA for the TCA cycle. When yeast ferments in a low oxygen environment, it is simply called fermentation. If it ferments in the presence of oxygen, it is called the Crabtree effect. The Crabtree effect and non-aerobic fermentation are distributed among the population of yeast strains. Further, the expression of genes for the variety of sugar substrates is differentially distributed, much like traits within humans are differentially distributed amongst the wider population. It is important to understand the wide variety of sugar substrates commonly found in fermentation. The two most common monosaccharide substrates are glucose and fructose. These exist in a circular structure for reasons derived from chemical stability and enthalpy. These do not occur normally in nature as monosaccharides. Rather, most sugars exist as a disaccharide. For example, sucrose or table sugar is a disaccharide of fructose and glucose. Complex carbohydrates as a group are comprised of starch among other saccharides. These are long chains of disaccharides. This is known as a complex carbohydrate in dietary terms, due to the slow process in digesting it in the human GI tract. This produces glucose. This led to the recent change from complex and simple definitions of sugar to the overarching scale of the GI index. Much like humans, yeast cells must break down the complex and semi-complex carbohydrates into simple carbohydrates or sugars that can be easily metabolized. Generally in this form. As a long chain of sugar, it is unable to be utilized by yeast. This is similar to the idea of spaghetti. It is a long, thin noodle. Trying to eat it as a noodle is difficult and inefficient. It cannot be consumed unless it is wrapped up into a small bundle or chopped into small sections. There is a similar effect in complicated media like juice. This has many types of sugar present and extraneous other factors, cofactors, additives, and compounds, among others. This makes up to 70% of any juice-based wash, whilst ethanol has 100% sugar content. With no other cofactors or extraneous variables, data shows that yeast utilization of sugars is dependent on a number of factors. These are yeast volume, yeast strain, sugar concentration, sugar type, time, total volume. As seen here, glucose is consumed by yeast at a rate higher than fructose and sucrose. This raises the question of why yeast is selective. Yeast is selective in the complex sugars it metabolizes. This is largely reflective of the enzymatic capability of each yeast strain. The selected strains of yeast can break down complex sugars using the enzymes available. This particular mechanism of sugar selectivity was observed in 1847 by Dubrofont and in 1886 by Berquelot. This is seen in yeast strains defined as glucophilic, which favor glucose as a result of more efficient cell wall transport mechanisms, or fructophilic, which favor fructose-based sugar substrates. An example of this is beta amylase. Beta amylase breaks down amylose and amylose pectin into glucose. The more complex amyloid pectin is a branching complex sugar, and as a result, being broken down more slowly because of its larger and more complex structure. This allows the simpler amylose to be consumed and used more readily. This selectivity has been demonstrated in experiments. These involved glycerol cells unable to metabolize glucose, but after 2 hours, the culture was able to ferment glucose. This was due to a synthesized protein. Culturing the yeast in conjunction with a chemical that blocked protein expression prevented the use of glucose. This was then compared with a second strain that preferentially favored fructose metabolism. Culturing this yeast in the same conditions did not affect sugar metabolism via an enzymatic pathway. This meant that it was a surface protein on the yeast cell, which selected for sugar preference. Other experiments have shown that glucose uptake is twice that of fructose. This is across multiple media, from brewer's wort through to mixed sugar substrates. As described previously, inside the cell, the glucose molecule is phosphorylated by kinases that isomerize it to fructose 6-phosphate. Then aldolase and other enzymes affect this until it gets to the pyruvate kinase. This finalizes the breakdown sugar inside the cell and creates 2 pyruvate molecules. This covers the basics of the intracellular metabolism of sugar. but that procedure begins halfway through the process. In order to get to the stage of glucose metabolism, each glucose molecule must be isolated from the larger chain of saccharides. This is done via enzymatic activity. As yeast exists in a suspension of yeast and saccharides, the yeast cell releases proteases, esterases, lipases, glycosidases, glucanases, pectinases, and amylases. These serve to dismantle the structure of whatever the sugar substrate is. For example, rice is a combination of starch and protein. Separating the glucose-rich starch from the protein is necessary. The following enzymes work to do this. Proteases. Proteases are used to break down proteins, known as proteolysis, and hydrolyse peptide bonds. This frees up amino acids to then be used by the yeast cell. Esterases. Esterase is the overarching term which encompasses the hydrolytic enzymes that break esters into acid and alcohol when in water. One of these has been previously mentioned, acetylesterase. This breaks off the acetyl group from another compound. Esterase activity is relevant to brewing in its compatibility with pectin. The common variant is polygalacturonase, which esterifies pectin. Esterification of pectin releases methyl groups. As described previously, this is generally an undesirable outcome as it produces a toxic byproduct. Pectinase. Pectinase is secreted from the yeast cell. On doing so, it interacts with the outer part of the substrate, generally the pectin-rich cell wall of plant material. Here it breaks down the physical barrier and creates an entryway to the sugar-rich inner parts of fruit, lipases. Lipases exist to catalyze hydrolysis of triglycerides to glycerol and fatty acids. Unlike the esterases described, lipases act in both an aqueous and non-aqueous interface, meaning both inside and outside of the cell. As a result, industrial production of lipid-specific enzymes use yeast as a primary means of production. These lipases act via a number of different mechanisms. These include breaking of oxygen bonds, alkolysis, acidolysis, and aminolysis. In industrial application, yeast and other lipase-secreting microorganisms are being examined as a means of creating synthetic diesel fuel. This biodiesel is largely interchangeable with fossil fuel-derived diesel. Carbohydrates. Carbohydrates are among the most important of these extracellular enzymes, as they break down complex sugar into simple sugar. In fermenting, this is essential. These make the small sugars needed for energy, the most common of which are amylases. Amylases come in two varieties of relevance to yeast. These are beta and alpha amylases. The alpha amylase only works in alpha bonds of saccharides. It is found in mammal salivatory and other digestive secretions. The other amylases are known as beta amylases. These are a glycoside that works on starch, glycogen, and similar polysaccharides in the beta form. It is not found in mammals normally. It occurs in yeast, where it is particularly good at disassembling polysaccharides into maltose. Glucanases. Glucanases are another enzyme. This enzyme breaks down glucan from chains of glucose monomers. The breakdown facilitates glucose metabolism. As with amylases, these exist in alpha and beta form. This is further delineated by the use of either a 1,3-beta or a 1,6-beta hydrolysis for extracellular activity. The quantity of intracellular beta-glucanase of varieties of specialist yeast strains is 344-fold, and extracellular is 144-fold that of a standard baker's yeast strain. Alpha-glucosidases act on the 1,4-glucose bonds. In the simplest terms, these enzymes all work in a fundamentally similar way. It begins by a somewhat unique section, surface, or composition of the larger polysaccharide. This can be the chirality of the molecule, alpha and beta normally. This selects for the variety of enzyme used. The specific enzyme is then able to bind to the site of action. This may be an oxygen or disulfide bond, or even a double bond in a chemically unstable configuration. When this alters the chemical structure, this leads to a break in the chemical's composition. This either separates into two compounds, as with glucosides, or breaks larger polysaccharide chains into smaller, short sections, which can be further reduced, the latter being done by larger, more complex enzymes. This is the oversimplified version of enzyme activity, as you can see in this short animation. Thank you for watching. Please post any comments, questions, or suggestions you may have below.